In this short video, we are going to look at how we can measure rates of reaction. So we've already seen how we can calculate rate of reaction by looking at either the quantity of reactant that's used up over time or the quantity of product that's formed over time. But to be able to use those two equations, we need to actually measure something in a lab so that we know how much reactant was used or how much product was used. And that means that we need to know the names of lots of pieces of scientific equipment and glassware. So I'm going to take this opportunity now, before we start looking at how we can alter rates of reaction and how we can speed reactions up, to actually stop and have a look at the equipment and make sure that we know the names of all of the different pieces and the functions that they are carrying out. So your keywords for this video are reactant, which is your starting materials, product, which is the chemical that's formed in a reaction, measure, which is obviously because we're trying to measure the rate of reaction, we need some data, we need a variable that we can measure. Fixed time intervals, because we need to be able to measure things regularly at set times. So fixed time intervals could be every 30 seconds or every 60 seconds, or every 5 minutes. It's just a good term to use in particularly practical exam questions. Balance, and in this case I mean top pan balance, which is what you'd probably call scales. So they're used for measuring the mass of a solid, but balance is a better scientific term. And gas syringe, which is a piece of equipment that you may not have seen yet, that's used to measure amounts of gas, quantities of gas. So for the next 6 slides, I'm just going to show you some different types of equipment. We're going to make sure that you can name them and know what they do. So a quick reminder, we need to be able to detect the quantity of reactant being used up or the quantity of product being formed over time in a lab so that we can then calculate the rate of reaction. Right, so here's our first setup that we could use in a lab to help us to detect the quantity of product being formed. So in this example, we have got all of these pieces of equipment. You need to know what this is. You need to know that one. This one's obviously labelled rubber tubing. This one is your gas syringe. You need to know the name of this piece of equipment as well. So take a few seconds, see if you can remember what all of these pieces of equipment are called, and then I will label them for you. So starting over on this side, we have got the clamp stand, which is what most students call it. It's also called a retort stand. At the top of the clamp stand, we have got the clamp, which holds the gas syringe, and that is connected to the retort stand using what we call a boss. That's the thing with the 2 wing nuts on it. So as long as you know clamp stand or retort stand and clamp, probably okay with that for now. Next we've got the gas syringe. So these are usually made out of glass and they're like really, really big syringes. And you start with this plunger pushed right in, and then as the reaction takes place and gas starts to be released, the gas will travel down the rubber tubing into the syringe and it will push the plunger out. So this plunger will start to move out over the course of the reaction. And when that reaction stops, or after a fixed time interval, you can have a look at where the plunger's got to and you can read off on the measurements how much gas has been produced. So this gives you a value for volume of gas produced. So we've got the gas syringe connected to rubber tubing, and the rubber tubing goes into here, which is like a rubber stopper. This is called a bung. And the bung is there to seal this. You should know what this is called. The shape should give it away. This is a conical flask because it's cone shaped. And this is where your reaction takes place. So inside the flask, inside the conical flask, we have got some reactants. So this could be an acid and some marble chips reacting together to give us some carbon dioxide gas, which is going to travel down the rubber tubing into the gas syringe and push the plunger out so that we can measure the volume of gas produced over time. And in the corner, I've got my little clip art stopwatch. So we need to measure this over time so that we know how fast it's happening. Now this particular setup is really, really important because this topic has required practicals, and one of the required practicals is to measure the rate of reaction and investigate how it changes when you are creating a gas. This reaction produces a gas. This could be one setup that you might describe in a method if you're asked to write a method for this sort of reaction in your exam. So make sure you're very clear on what all of these pieces of equipment are called, what they do, and how this particular setup can help you to measure the rate of reaction. Here is another practical setup that you could use in a lab, and this one also detects the volume of gas produced over time. So again, take a few seconds, have a look at the equipment, see if you would be able to label it. So this piece of equipment is the same as the one in the previous image. This one is a conical flask. And same as before, it is stoppered at the top with a bung. So you should know those. Now we've also got, going from the bung, we've got a delivery tube. So this delivers gas from one place to another. And these are usually made out of glass. The delivery tube takes the gas down here into a trough of water. Change my handwriting a little bit there. That's better. So a trough filled with water. So we've got water in there, just normal water. And here we have got an upside down measuring cylinder. So actually this would probably look a little bit better if it had a base on it. So it probably looks something a bit more like this. So this is a measuring cylinder and it's upside down. And you have to be aware that it's upside down because wherever this line is, you need to remember which way the numbers are going so that you can measure it carefully and precisely. Okay, so that's all the equipment, so you just need to understand how it works. So we've got in here your reacting materials. So these are your reactants. This is the only place where you've got the reaction actually taking place. This is all for detecting the volume of gas produced. So let's label that. Okay, so your reaction's taking place, and this particular reaction produces a gas. So that gas travels up through the delivery tube, down to here, and it comes out underwater, and that's what this water's for, because when the gas comes out under here, it will produce bubbles. So to set this up, what you would do is you'd take your measuring cylinder, fill it to the brim with water, and then put your hand over it, and then you would slowly lower the mouth of that measuring cylinder down into the bowl of water so that it stays filled with water right to the top. Then when the reaction starts and the gas travels down, the bubbles will start to push the water down. It will displace the water out of the measuring cylinder, and that water level will start up here and will start to move down. So now it's here and it will continue to move down. And using our stop clock or stopwatch at fixed time intervals, you can measure where that water level has got to, and that will tell you how much gas has been produced, the volume of gas that's been produced, and you can record that in a table. We will have another look at exactly how all of this equipment is used, as it would be for your required practicals in later tutorials, but for now, as long as we know how it's set up, what each piece of equipment is there to actually do, and what it's called, then that'll do for now until we get on to the next tutorials. Right, number 3, this is another setup that's really good if your reaction creates a gas. So here we've got stuff that's already labelled, so trusty conical flask. We've got a top pan balance, which you might call scales, but it is a balance, that's the scientific name for it. So we've got a balance that's measuring mass, and we've got a cotton wool bung, and here is where the reaction takes place. Now, this one does come up in exams and people do get it wrong, so you need to look very closely at what's going on. So the reaction's taking place in the bottom of the conical flask. It's producing a gas, and that gas rises, and this is where people go wrong. The cotton wool is not there to stop the gas from escaping. It's there to allow the gas to escape and to stop anything else from escaping. So the gas that's being produced can get through the cotton wool. Obviously, we should expect that gas can get through. So the gas can escape from here, but nothing else can. So none of the liquid, if anything's bubbling or spitting or anything, nothing else can escape from there. And as the gas escapes, we can expect that on here, we're going to see some change in the mass of whatever's in the flask. If the gas is leaving and the gas has mass, then we would expect the numbers on the balance to be decreasing. So we can, at set intervals, at set time intervals, we can see what the mass is and we can watch it decrease over time as gas is escaping, as the reaction proceeds. And like with any rate of reaction set of data, you would expect that over time, you've got a lot of gas escaping, so the mass would be quite high to begin with, and it would start dropping quite quickly and then slow down, slow down, slow down until it stops. And that's because the reactants are running out. So check that you understand what everything is in this picture and what everything does, and please don't make the mistake of explaining that the cotton wool is there to stop the gas escaping, because it isn't. Cotton wool wouldn't be good enough to stop a gas escaping. Cotton wool is used so that the gas can escape, so that the contents of the flask get lighter over time, and we can measure that. Okay, now we're going to look at two slightly different methods. So these methods are not for when you're producing a gas, these are actually for when you're producing a solid precipitate. So in the flask, we have got your reactants in here, and underneath the flask on the desk, we've got a piece of paper with a black cross drawn on it that's just been placed underneath the flask. So in terms of equipment, this one doesn't need an awful lot. We've got a piece of paper with a cross drawn on it, and we have got a conical flask. The reaction takes place here, so you'd probably put one of your reactants in and then get ready with your stopwatch as you're about to add your other reactant, and as you add it in, you would start your timer. And what you're looking for in this practical, because you're producing a solid in the bottom of the flask, eventually you'll produce so much solid that you won't be able to see the cross anymore. So you're timing how long it takes for you to no longer see the cross. So it doesn't directly tell you exactly how much product is formed over time, but it gives you a way of measuring something related to product formed over time. And this is quite good for comparing two different reactions, or reactions where you've changed something about the reaction, so that you can see which one's actually happening faster, because it's which one takes the least amount of time for the cross to disappear. So this particular practical is also a required practical, so there are two in this topic. One is for measuring amount of gas produced over time to give you an idea of rate of reaction, which are the three equipment setups that we've already looked at. The other one you have to do is to measure one with a solid, where a solid's being produced, and this is the prime example. This is probably what you would use in a lab to be able to do that. Now when you're looking at this practical, another word you might come across is turbidity. So turbidity is a measure of how opaque a liquid is becoming because of all the particles that are suspended in it. So your reaction mixture will start off completely transparent and see-through, so it will have very low turbidity, and as the reaction goes on, as time goes on, and the particles keep colliding successfully to react, the turbidity will increase. So that means it will get it'll go from being transparent through translucent to becoming quite opaque, so that you can't see the cross anymore. So that's a word you might come across. So you're essentially measuring turbidity, but you can just describe it as how long it takes for the cross to disappear. Finally, we've got one more method, which is very similar to the one with the disappearing cross, with a piece of paper underneath the conical flask. This one does the same sort of thing, just looks a lot more complicated. So we'll go through and label all the equipment first. So if you want to take a few seconds, have a look at the diagram, see how much of it you would be able to label, see which pieces of equipment you recognise, and then we'll go through and I'll tell you what all of the pieces of equipment are and what they do. So hopefully you've recognised where our reaction is taking place. This is a conical flask, cone-shaped conical flask. This one we've used previously in the one for collecting gases with the delivery tube into the trough of water. This is a measuring cylinder. This one you probably recognise from a medical perspective, from the doctors. This is a syringe. And we've got some light shields either side. So this is our light source. So this is a lamp. You can call it a light source. Hopefully you know what this is. This is a tripod. And here we have got our retort stand, boss, and clamp. And we've got one more bit of equipment that you might not be able to quite recognise. Here underneath the tripod, being held in place by a clamp from the clamp stand, from the retort stand, this is a light sensor. So have a good look at the equipment, make sure that you understand what everything is. These pieces of equipment, the measuring cylinder and the syringe, they're our reactants, or they contain our reactants. So we've got reactant here, we've got reactant there. You could put one reactant in from a syringe if you need to measure it very, very precisely, and then you could add something else using your measuring cylinder. So I wouldn't worry too much about the measuring cylinder and the syringe at the moment, because you could use those to measure out your reactant, so you could use something else. So let's just focus on the reaction once we actually start it off, where we have the conical flask, some light shields, a light source, a light sensor, and a stopwatch. So let's assume we've put our reactants into here, the reaction's begun, and we've quickly started our stopwatch. Now this is very similar to the one with the disappearing cross, so where you would have had a piece of paper here with a cross on it, and you're waiting, if you look down the flask, for that cross to disappear. This is just doing it for you, and this can give you a range of values, so rather than just waiting for the single moment when that cross disappears, this can give you a whole range of values. So you shine the light source down through your reaction mixture, and that light is detected using a light sensor, and you can imagine that to begin with, quite a lot of light is going to be travelling from here and actually reaching the light sensor. So to begin with, we're detecting loads of light coming through the reaction mixture and hitting the light sensor. So we're up here on our graph. Now this particular reaction produces a solid. So as the solid is being produced, it starts to absorb and reflect some of the light that's coming through from here. So not all of it is going to reach the light sensor, which means that over time, the amount of light being detected is going to drop and drop and drop until eventually the solution becomes opaque, because we've got such a high turbidity, we've got so much solid floating around in here that the light can't actually reach the light sensor, so it starts to detect very, very little light at all, if any. So this is exactly the same as the disappearing cross reaction, but it actually gives us a little bit more detail, because here we can actually look at any one time how much light is being detected, whereas with the disappearing cross, you're using your eyes only, and you're just looking for one particular moment in time where the turbidity becomes too high for you to see the cross. So that's it for my 5 different ways of measuring the rate of reaction. Hopefully you've had a good look at equipment and you've made sure that you know the names of all of the pieces of equipment, and hopefully you've got some idea of how you could go about measuring rate of reaction in a lab. So we're going to look in the next tutorials at how we can start altering the rate of reaction, make it go faster, make it go slower, in which case you would need something like this or one of the other 4 methods to be able to compare 2 reactions and see which one's faster, and then we can use collision theory to talk about why one reaction's faster than the other. So now if you could head over to the quiz, we will have a little look at what you remember about all of these different pieces of equipment.